our first step is the binding of the foreign bacteria. It means, let's draw this huge ether macrophage. It has nuclei. Let's call it phagocyte. It's a cell. It has nuclei. Nucleus, and also among many important organelles, I put most important for phagocytosis, your lysosome. It's an organelle inside this macrophage phagocyte. And what this phagocytes is famous for, it has the super receptors expressed at the surface. These super receptors will sense any foreign bacteria, because bacteria cannot invade your body without being recognized. Bacteria has a specific markers at its surface, which these super receptors will, or super sensor will recognize immediately. And this, if it's your bacteria, let's do it smaller because we will eat it eventually inside these phagocytes like this. And this bacteria has a specific proteins, markers of the bacteria. It means bacteria cannot kind of sleep on kind of recognize. It has a special markers which will be bind and recognized by the phagocytes and bind to. It means as soon as bacteria with this proteins on the bacteria, immediately will be recognized by phagocytes, and the super receptor will bind. And as soon as you bind the bacteria, you kind of have a transition into the second step of the phagocytosis, which is the step that will start to engulf this bacteria. It means from here you have transition to here, then actually what happened, your macrophage, this is this nucleus, this is the lysosome, and here you have your super receptors bind to the proteins on the bacteria. And you actually start engulfing bacteria. It means what happened in the step 2, you start wrapping around this bacteria. You start, the macrophage start wrapping around this bacteria until those edges meet. And this, now probably a good time to explain what you find inside these lysosomes. inside the lysosomes, you find very aggressive chemicals, which eventually, as soon as you give them a chance, will destroy the wall of the bacteria. They are lysosomes, lysozymes, and they are reactive oxygen species. They will be important part of destroying, of lysing the bacteria walls. It means the next step from the second, it moves to the third step, which is then you fully engulf your bacteria you catch. Your macrophage wrapped around, these two ends meet, and now inside, and this is your nucleus, and now your many lysosomes are here. And inside you have bacteria still bind to these receptors. And these new organelles, practically you have the membrane wrapping around the trapped bacteria. And this you can consider a special organelle, you call it phagosome. It's fully engulfed. Its membrane merge around bacteria. And now you have these lysosomes. They start releasing their harmful chemicals inside this phagosome. It means lysosomes digesting practically bacteria. Because lysosome, they dump its content inside the phagosome. And after this comes the last step, in which we have important part you should not forget for the exam. I will emphasize what I'm talking about. It means after some times, after lysosome dump their harmful chemicals, lysozymes and reactive oxygen species inside, they start digesting. They start lysis of the bacteria inside the phagosome. And it comes to the next final stage of the fourth step, phagocytosis. Let's call it present antigen on its surface. You mean surface of the macrophage. What happened? It means this is your macrophage. This is your nucleus, and this is phagosomes with bits and pieces of the digested bacteria. you might feel happy you destroy your bacteria, but macrophages also has another name, antigen-presenting cells, because what macrophages is doing, they use special complexes here inside the bacteria, macrophages. We have special complexes, MHC2 and MHC2. This is major histocompatibility complex. And what's its complex doing? It's kind of attaches piece of bacteria to its complex. It's actually protein. This is complex protein. And actually it's moving at the surface, and there are millions and millions of these major histocompatibility complexes expressed at the surface of phagocytes, which just broken down this particular bacteria with the piece of antigen. It means here at the surface, eventually major histocompatibility complex 2 with a digested bacteria peptide. And now, this is present antigen on its surface stage as super crucial to activate your adaptive leg of the immunity. Because these macrophages, which with destroyed pieces of the ammunition of this bad bacteria, will enter your lymphatic system. And in the lymphatic nodes, they find among many, many B cells, those cells, B cells, which has antibody against this antigen, and will stimulate T cell and B cell to stimulate this particular B cell to start maturate into the plasma cell and producing many, many antibodies against this particular antigen, against this particular pathogen. It means when you draw the diagram, don't forget this is super important stage. It's not the end of the story that you simply destroy the bacteria. You need to report to the adaptive leg of your immune system what type of foreign invaders are here and what type of antibody we can start producing. And how on earth antibody works? Somebody has a question? Okay, let's talk a little bit how antibody works. How antibody increasing this phagocytosis. This first type of phagocytosis, which happened as a first line of defense before you have antibody against this bacteria present, is kind of slow. As soon as you have specific antibody released into the bloodstream and arriving on the site of inflammation, this antibody will bind to the bacteria, specific bacteria. And for the phagocytes, this coverage, it's like you put a jam on your toast. It's very, very yummy for the phagocytes to have bacteria covering with this antibody. And this desire of phagocytes for this yummy jello that's increasing tremendously the speed of phagocytosis. It means antibody kind of binds into bacteria, specific bacteria, and now phagocytes really can bind this bacteria covering this yummy antibody and start phagocytosis much more faster. If you have antibody against viruses, what they doing, viruses, you have binding of viruses, antibody, you simply immobilize these viruses and prevent these viruses to enter your cells. It means how antibody works, why do we need antibody? Simply, they became more visible, these foreign invaders, viruses, and bacteria, and speed up tremendously phagocytosis. Any questions? Let's see how we're doing this time. We have 10 minutes left, right? I would like to present maybe a little bit of my slides.